Hey guys.
Hey, how's it going?
How are you guys?
Yeah, not bad. How are you guys?
Good, good.
Hello.
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 Just to let everyone know, I think I automatically set to record this session, and I guess it would be helpful later on, also just for us to rewatch what's going on, the discussion that we have in this session. So, do we all give consent to that?
That's fine with me.
Thank you. No problem with other people, I hope. All right, I guess that's a yes.
Yeah.
All right, thanks. While waiting for people to join, I'm just gonna share the screen that the PowerPoint slides our team made already. And we're gonna start with a very brief update of what we are up to, and then going into some questions, if that's okay with Tyler and Christy.
Sure, sounds good.
Okay. Thank you. So let me know if you guys can see this.
Yep.
Yep. Yep.
So currently, we are looking at the 2 stages of configuration. And last time, I think Tyler and Christy both suggested that the first stage we use to consider this stacked design.
Mm-hmm.
So this is what we are thinking of right now. So this will probably be our initial stage for next year's team to be, and then later on we could move into this more complicated but also probably more efficient concentric tubular design, right? Though another question is, we are not sure how complicated it is to have the stack design, and would it be more straightforward if we just start with an H shape, or do you guys think it's okay to also start with a stack, and it wouldn't be too complicated for us?
So you may want to start with the more simplified design. You know, you can have the goal of expanding it. Typically, you know, it's easier to troubleshoot if you have, you know, fewer chambers that you have to kind of see what's going on with. So I'd recommend the more simplified design initially, and then if that is working well for you, then you can go towards the more stacked design with the additional cells. But you know, of course, that's up to you. It won't be difficult as far as construction. So construction either way would be pretty simple, you know, pretty straightforward, I would say. But as far as like, you know, sometimes you run into kind of unexpected problems or things that, you know, you didn't anticipate, and a lot of times it's easier to troubleshoot if you have fewer chambers involved. So that would just be the only recommendation that I'd have, but both designs would be, you know, appropriate for this.
Okay, sounds good. Thank you. And just to do a quick recap of how many chambers we're going to have. So last time, I think we were talking about having all of our species. So we are going to have 3 species, E. coli and P. putida for plastic degradation. And then our exoelectrogen is going to be Shonella. And last time we were saying to culture all of them under anaerobic conditions because that's going to optimize electricity production, right? However, from our modeling group, we found out that E. coli and P. putida, they need to be co-cultured under aerobic conditions. So that's definitely going to change the configuration. No matter whether we go with H-shaped, stacked, or concentric tubular, it's going to change drastically, and just we are not very sure about that. I just wanted to have your expert opinion.
Yeah, so that does make it a little bit trickier. So I'm trying to think how you might be able to do that. Now you could possibly have these microorganisms in a, and I don't want to add too many chambers to your system, but you could possibly have them in an exterior chamber, and it depends on the level of oxygen that's going to be required. So when you have, if you were to put them, oh yeah, it looks like, yeah, you already have that idea there.
Yeah.
That's great. Yeah, so the only issue there would be making sure that when you bring in the liquid from that oxygenated chamber into your anode, that there's still going to be oxygen, dissolved oxygen carried with it. So, I guess you could try to figure out, for example, what's the minimum amount of oxygen or dissolved oxygen concentration that you can grow those with. Because any little bit of oxygen that you do bring with the liquid into your anode, that will reduce your exoelectrogenesis, or your, efficiency at your anode. So, there are ways, you could, potentially do a step in between to deoxygenate. So, people, sometimes you could bubble through like some nitrogen or something like that to, remove some of that dissolved oxygen concentration that's there. But it also depends if you can grow those species at really low oxygen concentrations, then it may not be as big of an issue, and you may be able to just accept the loss of efficiency in the anode. So that's something to kind of consider. I know most of the time, when we consider something aerobic, it's around 2 milligrams per liter of dissolved oxygen or above. And so I know some microorganisms can grow with microaerophilic conditions. So I don't know if that's the case for these species. But if you were able to do that in kind of microaerophilic conditions, then I would expect that you're not going to have a whole lot of excess dissolved oxygen there. The other thing to think about too is if you're going to be growing them on a biofilm, or whether you're going to be growing them in suspended growth. So sort of like a mixed culture in the oxygenated chamber. So I think what were your plans, or what were you originally thinking as to how to set that up?
So we did come across that, and so our project was like, originally we were planning on feeding pure PET as you come back from Sigma Aldrich, although that is quite expensive.
Mm-hmm.
So as an alternative, we were thinking of just growing the cells as a biofilm on top of plastic bottles that we can collect ourselves for the iGEM project. Obviously, the plastic bottles would have to be treated before putting them with the bacteria. As another option for measuring the degradation of the plastic bottles, we were thinking of using a scanning electron microscope and take a scan before and after the degradation of the bottle. And I'm sure we will see some degradation happening on the surface.
Mm-hmm.
But then the scanning electron microscope is also expensive, so we are kind of stuck with that. The other option would be kind of, I don't know, somehow getting a batch in the O2 chamber with the two species, where they are degrading the PET polymer, as well as generating a lot of lactate that can then be transferred to the anode.
Yeah. So I think, yeah, a couple of things. If you are going, I kind of agree with the suggestion to do sort of a batch fed system. So the reason for that would be you could lower your oxygen, your final dissolved oxygen content to the lowest possible before transferring into your anode. And you can do that even with a biofilm or, with suspended growth, whichever. The other option to think about is with a biofilm, depending on how thick that biofilm is, there can be some mass transfer issues as far as oxygen getting to the biofilm that's more towards the interior of the biofilm that's against the plastic. Um, so, if you have a really thick biofilm, you're going to need a higher dissolved oxygen concentration in the bulk liquid to be able to drive that oxygen to the microorganisms that are actually at the plastic surface. So, in my, and I haven't done, much work as far as biofilms on plastic before, but I, just from, what kind of I'm intuiting, I guess, I almost think that it might be worth it to do sort of a mixed culture, a suspended growth culture that's continuously mixed. And you have small pieces. So you would take a bottle and you would, break it into small pieces or crush it, or, shred it basically, so that there's a higher surface area and the biofilm that would form on those pieces. So even though you might have it continuously mixed so that it's all suspended growth, that growth can happen on the little tiny pieces of plastic, but it's not going to form a really thick biofilm because you've got it continuously mixing, plus you have a lot of surface area. So they would preferentially be forming a very more thin biofilm rather than a thicker biofilm. And that would also help with reducing any sort of mass transfer issues as far as oxygen getting to the microorganisms. So I think, that would be what I would recommend, thinking about, shredding a bottle. And of course, you can still do, scanning microscopy with that. You could take an initial piece and do scanning microscopy, and then you could take a piece after, biofilm colonization and things like that, and then take a look at that and see how that's degraded. So, I think maybe shredding and having it be kind of a continuous mix system, and then maintaining that oxygen concentration, maybe putting in enough oxygen that you know that by the time the batch run is done, that you'll drive that oxygen concentration down so that it's relatively low before you transfer it over into the anode chamber. And you could do that. So you could do that a couple ways. You could supply it with enough oxygen for a period of time. So you could do aeration or keep it open to the atmosphere. And then at a certain point in the batch feeding, you could close it up and then allow it to drive that oxygen to lower levels, so that when you do transfer it, you don't have as much oxygen. The other thing that you could do as well, at the end of, say, a batch feeding cycle, you could just bubble in a little bit of nitrogen, or bubble a little bit of nitrogen through that system. That'll strip out a lot of the oxygen as well. And so that will, allow you to reduce your oxygen even further, and then you can, transfer it over. So I think a batch fed system is going to be the most optimal for this. And, I think that by having it kind of in that separate chamber, you can do a lot of things to make sure that you're not, bringing a lot of oxygen into your anode.
Okay. So just to make things clear, you would do it like in a two-step process rather than a whole apparatus where the O2 chamber is connected to the anode, right?
Yeah, so I would do it, yeah, like a two-step process so that you know, do your plastic degradation and your lactate formation. You may consider if, you know, if you have an oxygen probe available, you could even use that to measure the dissolved oxygen in there. And if it seems like it's relatively high, you may just bubble through a little bit of nitrogen to strip some of that out, and then transfer the liquid into the anode. So yeah, it would be kind of a two-step process, not necessarily like a continuous flow system.
And how would you get rid of the two species that were originally in the O2 chamber before transferring them into the anode?
So you could do some filtration. You know, I think, you know, that's, you know, that might be something that you could remove it, filter it, and then bubble maybe a little bit of nitrogen through it just to get any you know, oxygen out that came and dissolved during the filtration process, and then transfer it into your anode. And you know, even doing it, even though it, you know, even though you're doing it like that as a two-step process, taking it out, doing a few treatments in the lab, it doesn't negate the value of having this kind of a process. So basically, a lot of times we have to do those things in the lab, but if this was to be adopted as like a larger system, you would have online things in place where, you know, you would have a filter, you would have, you know, maybe you know, a deoxygenation chamber, and you would have all of those in place. And so what you're doing is really just mimicking that, but on a lab scale. And so I think the easiest would be to do your batch system in the aerobic chamber, take it out. You would want to filter it through a 0.2 micron filter. And you could retain some of that biomass even from the filter and rinse that filter back into your system so that you're not losing a lot of biomass. And then you take that filtrate, you deoxygenate it by bubbling a little bit of nitrogen through it, and then put that into your anode, and that should be carrying your lactate from the plastic degradation in there. So I would think that that would probably be the best way to do that if you wanted to maintain, you know, the biomass in that system without transferring it into the anode.
Okay. So yeah, so you said that then you would recover the biomass from the filter, put it back in the O2 chamber, keep the process going, right?
Right,
And that
yeah.
ensures that, and that would ensure that lactate would be transferred to the anode chamber.
Right,
Yeah,
right. So the lactate, because it's soluble, should go through the filter. The biofilm or the biomass should, in any sort of you know, remaining plastic pieces should be retained on that filter. And then you could put that, you could rinse that filter back into your system. And so, you know, you could do even like a more coarse filter first, and then that would remove a lot of the large plastic pieces, things like that. The other option there too is to do centrifugation. So instead of filtration, you could do centrifugation. So you take and put this into centrifuge tubes, you centrifuge enough to kind of form a pellet at the bottom, and that pellet would have the plastic and the microorganisms, and then you just take the liquid from, you know, the top part of the centrifuge tube, and then you transfer that. So, you know, it depends, you know, what equipment you have available. With the filtration, you would need a vacuum filter. So it, you know, kind of depends on what you have available. I think either of those methods would work. Now that I'm thinking about it, I would almost recommend the centrifuging method over the filtration, just because it's a lot easier to resuspend that pellet in and put it back into the system. So with the filter, you can rinse it, but there's still going to be some material that's stuck on that filter, whereas with the pellet, it's usually fairly easy to resuspend it by adding more liquid, more medium to it, you know, shaking, and then it resuspends and you can put it back into the system.
Okay. So you wouldn't really need a filter for the centrifugation step.
Right, right. Yeah. I mean, you will be carrying like maybe a few cells that are still suspended, but I don't think that that would be an issue at all.
Yeah, okay. Well, that was great. That was my main question, and you answered it amazingly.
Great.
Is Pedro? Did Pedro have another question, Anna?
Yeah, actually, I have a question. So, Christy, if we use centrifugation to like retrieve bacteria from the aerobic chamber, would that cause a huge damage to the bacteria, like cause lots of deaths of bacteria?
There will be some based on just shearing, but if you know, I wouldn't expect you to really need very high RPM, you know, speeds to be able to centrifuge it down. So especially if the microorganisms, if they're typically using the plastic pieces, they're going to be very much associated with those little plastic pieces. And that will also help you be able to easily centrifuge them out, more so than if they're a really suspended and you know, individual cells floating around. So so I think there will be some, but I wouldn't expect it to be very high. I would say use the minimum speed possible that you would need to be able to remove the, you know, the majority of the cells, and you know, you can do that just kind of visually almost. You know, start off with a slow speed for, you know, a short amount of time, and then, you know, see if you're forming a good pellet, you know, then that's great. But if not, then you can continue to kind of either keep the speed low and increase the time, or have a little bit higher speed and keep the time low. So I don't, you know, there will be some death, but I don't think that as long as you're not centrifuging at really high speeds and for a really long time, I would expect them to mostly be viable afterwards.
Yeah, okay, thank you.
I did have a quick question. I think the top one just there on this slide. So for example, like the initial experiments such as co-culturing or measuring Pété's activity or lactate production, would it be easier to conduct these in a system less complex than MDC, for example, just an acrylic sort of box really with co-culture active, rather than straight into the MDC?
Yes, yeah, so I agree. Yeah, I think, you know, you could even do it in, you know, like a bottle, for example. You could do it in a serum bottle, like a large enough serum bottle where, you know, you could put in your system. You could actually do it in Erlenmeyer flasks, for example. So you could do a number of different, you know, things, but I do think that, yeah, just having it in kind of a separate, more, you know, easily developed type of a reactor, or, you know, a container, and then from there you're going to be removing it anyways, doing centrifuging, and then, you know, possibly bubbling with nitrogen and then putting into your anode. So I think you don't really need those to be connected necessarily. Was that your question, or did I?
Yeah, I think that was slightly more towards the, like the late, once the idea is more fleshed out and developed. To keep this. Okay. I was asking sort of like just for the very initial first experiments, would it be easier, instead of bothering to build, before we built, for example, a prototype of the MDC, just to use a more simple system, like
Yeah,
at the very first steps?
absolutely. Yes, definitely. Yeah. So, you know, the more simplified you can make it. So yeah, for that one step, you know, you don't really need to build the, you know, the anode, the cathode, and that kind of thing. Really, what you can focus on first is trying to make sure that in that system, your microorganisms are able to break down the plastic and produce the lactate that you're looking for. And so really you don't need to have that entire other system built to be able to do that stage. Yeah.
Okay, great, thanks. I think, yeah, and then the second question was answered before, about using the H-shaped simple configuration as the first one.
Yeah.
I'm not sure if the third one has been asked, but yeah, it basically just says whether, like for example, the concentric tubular configuration that we showed earlier. One of the first things that I think you said to us before is that it's quite hard to implement the membranes, given that they're normally quite rigid, and to make them into cylindrical shapes quite hard. And then also like just the space of each compartment, whether that would be enough to do all the things that you want to do. Obviously, we've now talked about separating the PET degradation and lactate production. But just wondering how feasible you think separating those two things are with the concentric design that we would be aiming for.
Yeah, I do think it's feasible. You know, it's a good question that I don't necessarily think that you need to answer all of the questions about what the best configuration would be. So this is I mean, as it is, it's a very you know, what's the word? It's very like you know, great idea that's got a lot of pieces to it, I guess I should say. So you know, you want to simplify it as much as possible, and there are going to be some things where you're not totally optimized, but I do think that you know, if you're, you know, trying to show what your aim is, and this is, you know, where you're going with the system, I think that, you know, it is possible to still do the concentric tubular design. So, you know, but it may take a little bit more of kind of, you know, some engineering thought to how that would, what it would look like exactly. So I think, you know, my thought is that you can have kind of a, like a proposed design and say, you know, this is what we're proposing and this is where we're trying to go, but you don't have to necessarily have all of the little details worked out as to how that concentric tubular design would work, at least at this stage. So so I think, you know, yeah, so here we go. Exactly. So yeah, I mean, you can show, for example, like the exterior chamber and having it pumped into the anode in that tubular design. So you're feeding it with the lactate from an external aerated chamber. And I don't think that necessarily at this stage you would need to resolve all of the engineering issues that might arise with something like that, because that in itself, I would say, is kind of master's level thesis type of work. So, you know, I could see putting a master's student on that project just to try to like develop a system like that. So, so, you know, very, it's always a great idea to have that as an idea, but I don't think necessarily you need to solve all those little engineering problems as far as, well, how is it going to be pumped in? You know, how, I think if you have kind of a process flow diagram of how you envision it, then, you know, these other things can be worked out from there. Like, for example, how do you, if you're pumping in into the tubular system, how are you going to de-aerate? You know, you don't necessarily have to have maybe all of those things. Now, the other thing too, with this tubular design, so one thing to think is that if it is a continuous flow system, so for example, I see, you have your analyte being pumped in at the bottom, and I'd imagine being pumped out at the top, what's going to happen is sort of what we call a plug flow system. And so what that means is that it's not the same concentration of dissolved oxygen all the way through. So what you would have is where it's pumped in at the bottom, you're going to have a little bit more dissolved oxygen entering the system. That's going to be utilized by whatever microorganisms are growing in the biofilm near the bottom. And then as it travels up through, that dissolved oxygen concentration is going to decrease. And so by the time it gets to the top, you should start to have more efficiency up there as far as exoelectrogenesis goes, as long as you have enough lactate still available. So, so I think there's a lot of issues there, but I think just having that as your goal and showing a good process flow diagram, I think at this stage is going to be plenty to show that you have this vision of where to go with this.
Okay, great. Yeah, I was thinking something like, given that we talked about separating the two processes now, I was thinking also that even in the more simple H-shaped configuration, that would still be a lot of work for next year's team to figure out all the co-culturing, everything else. So yeah, I guess that should be the plan then. Just have it as a proposal and how we would attempt it, or how maybe next year's team would attempt it, rather than having to have it all figured out. Okay, thank you.
Mm-hmm.
I think there's a couple more questions on that slide, but
yeah.
I think they were Anna's questions.
Yeah, so another question was, from the process of plastic degradation, we are going to generate ethylene glycol, which is toxic, and just considering the configuration, how would you envision it to be removed from the chamber?
That's a great question. Yeah. I'll have to think on that one a little bit more. I don't have an answer right off the top of my head for that one. Yeah, have you, now I'm wondering, is that also known to be toxic to the Shigella? Have you seen that in literature?
We are not sure whether it's going to be toxic to the Shigella, but for E. coli and P. putada. So like the very beginning, we were thinking that E. coli could uptake ethylene glycol as its carbon source and grow solely on ethylene glycol as a result of PET degradation. But then later out, we found out that we probably also need to supply glucose, and then E. coli might not be able to uptake ethylene glycol as we expected in the very beginning. And that's why we're thinking of having to remove ethylene glycol from the chamber instead of using it for biomass growth.
Yeah. So,
but is it toxic, Anna? What did you find? Is it? Ethylene glycol is just a sugar. What would it be?
That and it says that it's toxic.
I think it's toxic, and also ethylene glycol is soluble. So if we cannot extract ethylene glycol from the liquid that we are going to pump into the anode chamber, that could cause a huge problem.
Yeah, I think I'd recommend trying to look at the literature a little more to see if there's any studies on the effect on its effect on Shigella in particular. So, sometimes, something that's toxic to one species might not be to another. The other important thing would be to understand the mechanism of how it's causing toxicity. So for example, you know, if it's like disrupting the cell membrane, for example, you know, that would be a lot more difficult to get around, I guess, or to deal with than if it's something that's more specifically uptake, you know, oriented. So, you know, a transporter or something like that. So I think that would take a little bit more research on what exactly is happening. So it may be, and the other point that I, you know, to make there is to figure out what concentrations you would expect of the ethylene glycol if the degradation goes as expected, or as, you know, based on how much, you know, if you have good PET degradation and you produce so much lactate, how much ethylene glycol would you expect to be in the system? And then because a lot of times, especially if there's biofilm growth on the little pieces of plastic, the biofilms themselves can be somewhat protective against toxicity. So if that is an issue, then you know, we might have to rethink how, you know, whether you may want to have a more thicker biofilm, you know, in your system rather than the thinner biofilm that I was recommending to kind of speed things up. But there usually is kind of this threshold concentration where below that threshold concentration, there's very little toxicity to a biofilm from various things. So I think it's going to be concentration dependent. And also to understand whether it's a reversible type of effect on the microorganisms. So for example, you don't, if it's something that once that ethylene glycol is removed, that the bacteria go back to normal and can, you know, go back to their usual metabolism, it might just be something, you know, sort of like, you know, where a lot of buildup of products from a certain metabolism can slow that metabolism down. And it's sort of the biological equivalent of Le Chatelier's principle, where when you form too many, you know, when you drive a reaction forward and you start to accumulate a lot of products, it starts to make it so that it almost thermodynamically wants to go backwards. And so so that can cause that biology and those metabolisms to kind of slow down and stop until that those products are removed from the system, in which case you may not have as much of an issue, because you can have a batch system that does slow down at the end, but then you remove that liquid, replace it with new medium, and then now they can go back to doing what they do, and, you know, getting back to their metabolic, you know, processes. But there are certain non-reversible toxic compounds where if you know, if it lyses the cell, for example, those cells are not going to be able to, you know, go back and start, you know, doing, you know, their metabolic processes, breaking down the plastics once you remove that. So I guess understanding maybe the mechanism of that would help to kind of better formulate how to deal with that, and understanding what concentrations you might expect from the, you know, the breakdown of the plastics from that. But that's a great question, and I really don't know the answer straight away from that.
I kind of want to add, I just put something in the chat right now, but I just looked it up right now, and it looks like there is an engineered strain of E. coli that actually degrades ethylene glycol. So you might want to like check that link I sent, because it might be available to purchase, or it might be available from the lab, like the lab that engineered it. So someone has it actually degrading ethylene glycol. So
Yeah, and also I found a paper just now where Pseudomonas can degrade ethylene glycol natively if you upregulate some genes in the pathway. And we have Pseudomonas already in the culture, so.
Mm-hmm.
But yeah, I'm assuming it would be more of a problem of accumulation over time
Mm-hmm.
rather than disrupting the membrane
Mm-hmm.
or things like
Yeah,
that.
I would expect that too. Yeah.
Yeah. With regards to the previous question that Lee asked, where he said that you can, if it's possible to recover the cells after centrifugation, utilize, like, sort of recycle them again. Can you grow them? Like whatever you recover, although some of them may die, can you grow them? I assume you can grow them again, right?
Yeah.
So yeah, so that would kind of compensate for the cell death that occurred during the centrifugation process, right?
Right, right. And yeah, you can think of it as, and actually we used a very similar process for wastewater treatment. You know, we do have certain centrifugation methods to remove biofilm or biomass from, you know, like an aeration tank basically, and recycle those to the beginning so that it allows it to reseed and, you know, re-inoculate, so you don't have to regrow the entire culture. And so that allows you to really maintain a certain amount of biomass in the system that, you know, you can keep it almost constant except for during your batch system or your batch feeding.
Yeah, okay. And also with regards to testing the system before, like on a small scale or outside of the microbial fuel cell even, how would you need to, in order to measure the amount of lactate being generated by the co-culture, would you need to purify that chemically, so chemical purification, or using column chromatography, or can you use a dirty sample taken from the batch?
So typically the way I've done lactate measurements have been through, well, first of all, I use high performance liquid chromatography, HPLC, and I believe a refractive index detector, RID, is required to be able to detect the lactate. And you need to make sure you have the right column in your HPLC to be able to separate it out. But those are usually, it's usually a pretty standard, I bet that, you know, you could find a configuration of an HPLC that would allow you to do that. So the preparation would be basically taking a sample of it. You wouldn't need to extract lactate or anything like that. You would take it within the liquid medium. And the only preparation really that I do is filtration through a 0.2 micron filter, and that removes the cells. And so that way those, you know, the microbial cells, you don't want to put those into an HPLC because that can clog the column up. So as long as you do a filtration, and if you are taking small samples, what I recommend for that would be a syringe filter. So basically you can take a clean syringe, just take it, filter it right into a little vial, cap it. You know, depending on your method, you may need to acidify. And in that case, you know, but it depends on the column. It depends on, you know, the configuration of the HPLC. But if you find a lab that does do regular lactate measurements, you know, they should have that procedure. It's actually a pretty common thing to measure with HPLC.
Okay, thank you so much.
Sure.
Yeah, thank you. We just got this one question, and I'm not sure which one of my teammates asked it. Someone explain this, please.
Yeah, so I was just wondering, so we have two separate chambers, and in different chambers, we are culturing different bacteria. So if these kinds of bacteria, they grow in different environments. During the process of lactate transferring, how can we ensure that the lactate we transfer from one chamber to the other and does not disrupt the culture environment of Schwannella?
Yeah, so what I would think would be the best method. So I would typically do a well-buffered medium that has a relatively neutral pH. So pH somewhere around 7, which usually most microorganisms like that. So I would expect both in the aerated chamber and in the anode that you know, pH around 7 would be good. And so if you create a medium that will work for both your anode bacteria and for the bacteria in your aerated chamber, then there really shouldn't be a whole lot of issue there. So what you might need to do is make sure that, you know, you're doing a well-buffered medium, and then you're adding certain vitamins and minerals. And so you may need to look up what medium is best for each of these microorganisms and make sure that you're at least supplying enough vitamins and minerals for all of them. So if, for example, one microorganism, the Schwannella, requires a very specific vitamin that the others don't really necessarily utilize, or a specific mineral, just make sure that that's in the medium for the aerated chamber, so that when you do that centrifugation, you're carrying over that medium and the lactate into the anode, and you'll have all those, you know, soluble components like the vitamins and minerals. And as long as it's well buffered, you know, usually like a phosphate buffer is great to use. As long as it's well buffered, your pH should stay relatively neutral. And, you know, I would expect that you would grow all of these kind of at room temperature, so that you don't have to add the complication of, you know, different temperature or different pH or anything like that. So I think, you know, the closer that you can make the, you know, culture environments of both, the better. So if you give it a really, really rich medium, you typically don't have to worry about, you know, not having enough vitamins or not having enough minerals. Usually I do like a you know, 10 times, a 10x media, basically 10 times what's usually utilized. And we can give you our medium recipe if you're interested, for our analyte. And you could always tailor that. If there's something missing, you could add that in, and then just use that for the aerobic chamber. And then when you centrifuge out the microorganisms, the soluble components will all transfer over into the anode. And you know, there shouldn't be a whole lot of issue there. Does that answer that question for you, or?
Yeah. So I guess if we are gonna do a two-step process, like even though the culture environment for the aerobic chamber might not be identical with the anode chamber, we can further process the supernatant coming from centrifugation to add some certain vitamins and minerals, and then put it back into the anode chamber. Because we actually use two steps, so that shouldn't be a big problem. We can.
Yeah, you could do that, or what you could do is just add those, you know, up front into the medium that goes into the aerobic chamber. And, you know, usually an overabundance of vitamins and minerals doesn't really harm the microorganisms too much. So, so you could add, you know, an abundance of those up front into the aerobic chamber, and then the supernatant should also carry those, you know, into the anode as well.
Yeah, thank you. And by the way, that would be great if you can share the recipe of the media with us.
Sure, no problem.
Well, I
use
guess
a very heavily buffered medium. So just to point that out, so you may not need as much of a buffer as what our medium utilizes. And the reason for that is when we have the medium that we have is kind of tailored for an anode that's attached to a biocathode where it's really active. And so we've had to buffer it very heavily to maintain a neutral pH. I don't think that you'll have that issue in a microbial fuel cell. So I don't think that you'll need that high buffer concentration. So you may be able to reduce some of the phosphate buffer concentration. But I just wanted to point that out. So when you do see our medium recipe, you can reduce the buffering a little bit there.
Yeah, great, thank you.
any other questions, everyone? Otherwise, I think that's all we got from the slides, at least.
I had a question. Do you all know when you're picking the next team leads for next year?
So when was the deadline to apply for iGEM mono? Was it like February, January?
Yeah, it was like the end of January, January 20th, something. And I think we usually got all the recruits to meet like what, late March, early April-ish. That's probably the time, and then they'll have to communicate amongst themselves to have a leader. So
Okay.
like I'd give it to be April, mid-April, most likely.
But I think you mentioned last time, or someone mentioned last time, that some of the members from this year's team might stick around for next year, or is that how it works?
So like, for example, I guess for my case, since I'm not yet graduating, and I'm not gonna graduate for a couple of years, so I might still stay in the department just to help out, and but I don't know whether I will be like full-time working on the project.
Mm-hmm. I'm writing proposals right now for San Diego State's iGEM, and we wanted to collaborate with some of our ideas and combine them with this idea that you all have been working really hard on. So I just want to make sure whoever is working on it next year, either if they're working on it or not working on it, that they're okay with us moving forward with some of our ideas on the project. We talked about it earlier and you all said it was okay, but I just want to check in again just to make sure everyone's like okay with us trying to collaborate with you.
Yeah.
So
It's fine.
okay. Sounds good.
Yeah, usually, at least at UCL, it is not common that the same students participate in iGEM twice, since they want to give the opportunity to other people as well, right? But usually the team is chosen end of January, February. We go to an interview, and then they just choose the students that will, create and form the team, essentially.
Okay, sounds good.
Yeah. And some of us will be still at UCL, but basically because they are second year, so they will be going into the third year. But others will be doing masters or something else. Yeah.
Yeah, and that also depends on how much they need us the next year's team.
Yeah. Okay, well, thank you. That answers my question.
So yeah, given that we don't have lab access at the moment, we are playing around with flux balance analysis to kind of estimate how much lactate we would be producing. When we'll give those outputs to next year's team, so they know where to start. They know they can look at our models and make decisions into how to actually perform the lab experiments. We are also modeling the biofilm growth on the anode, which only assumes Shonella at the moment. But yeah.
Well, awesome. If you guys don't have any more questions for us, I think
Yeah.
you answered all our questions too. So.
Okay. Thank you so much for joining.
Oh, sure. Yeah, you guys are making some great progress. So it's really interesting to see, you know, your work progressing, and you're doing a great job researching all this. So yeah, interesting project. I really like it.
Thank you. Well,
Thanks, guys. Take care.
Yeah,
yeah,
thanks.
okay.
Nice talking to
Thank
you.
you. Take care.
Bye.
Bye
Bye.
bye, everybody.
Bye.
Mm.